Welcome, everyone, to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Multiplexed IHC ISH using an automated slide staining platform. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Proper staining and labeling of biological tissue samples using either immunohistochemistry or in situ hybridization techniques has been a fundamental part of cell biology and histology laboratories for decades. However, preparation and staining methods have been notoriously time-consuming and until recently have been overlooked as high-throughput applications and equipments were being developed for other standard assays. Recently developed techniques and equipment have now allowed researchers to streamline their staining and label workflows, providing them with the opportunity to test an array of research conditions in exponentially shorter times with greater reproducibility. Let's meet our speakers who will describe for us their experiences and tell us about their methods while using an automated slide staining platform. Our first speaker today will be Adrian Murillo, scientist at Ventana Medical Systems, a member of the Roche Group. Mr. Murillo will discuss his methods for multiplex labeling assays using the Discovery Ultra platform. Mr. Murillo will be followed by Edward Abril, a research specialist at the University of Arizona Cancer Center. Mr. Abril will highlight his use of Discovery Ultra platform for multiplexing staining with the immunohistochemistry core facility at the University of Arizona Cancer Center. Before our speakers get started, I want to encourage you to submit your questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. With that, let's get our webinar underway. Adrian, you have our attention.
Thanks, Jeff. Today I'm going to be talking about methods for IHC multiplexing on the Discovery Ultra. Okay, so here's just a quick outline of what I'll be talking about today. But first and foremost, why multiplex? A lot of researchers multiplex for the reasons of just seeing if there are proteins of interest that are co-localized within the same cell. Is this cell expressing in these two proteins? Are they expressed in the same cellular locale? Oftentimes, researchers may want to interrogate whether or not protein expressions are different in relation to differing cell populations. For instance, is this macrophage expressing a certain biomarker in relation to a T cell? What is this T cell, neighboring T cell, expressing as well? It enables the researcher basically to interrogate, in the context of tissue, which cells are expressing which proteins. And also, in the past, multiplexing has been used as a sort of a makeshift proximity assay. And the fact that, in this cartoon schematic that we have here of these two made-up biomarkers cartoon scheme here, you have biomarker 4 and biomarker 3. If they are truly co-localized in direct proximity to each other, if you are using a fluorescent detection of a green and a red, you would see, if they were co-localized, you would see the two colors mix and form an orange fluorescent color. So this is one of the many reasons why to multiplex. But multiplexing does come with its pros and cons. At some point, how much is too much? At some point, you're going to put so much color on the slide that it's going to become very difficult to interpret. And at what number does that happen? Is it with 4 colors per slide? Is it 8? Really, that's up to the user to define for themselves. But at some point, you will require some sort of digital analysis to help you interpret the results in which you achieve with your multiplexing. Also, to just take into consideration assay time. With multiplexing, you are doing a lot of markers per slide, so the assay time can get lengthy, and it can go from anywhere from 2 hours to 48 hours, or maybe even more, depending on the type of multiplexing you're trying to achieve. And then, at your disposal, you do have fluorescent and chromogenic detections, and each one of those has their own pros and cons as well. With fluorophores, you are able to get them relatively cheap, and they do have the properties of when they co-localize, they do form a third color. However, fluorophores do have the property of photo bleaching, and so over time they will lose their signal, and therefore you won't be able to archive these slides. With chromogenic detection, that needn't be the case. You could store these slides; they're archivable. However, with current chromogenic detection technology, they don't really hold that property of a fluorophore of being able to co-localize and form that third color. So currently, the ability to multiplex is generally done in fluorescence, and it is a lot easier to do higher order multiplexing. 3 triple stains, maybe quadruple stains, or pentuple stains with fluorescence, just for the ability that you could filter out the signals and see which cells are truly lighting up with your respective fluorophore. Fluors do currently have a larger library to choose from, and just the property that I mentioned previously is the fact that when fluorophores do co-localize, they tend to generate that third color, so you can actually see co-expression of certain proteins. So right now, it seems that for multiplexing capabilities, it does seem that fluorescence is the route to go. Here at Ventana, our Roche Tissue Diagnostics, we actually do have some fluorophores which are conjugated to ceramides, so thus needing an HRP in order to deposit these. And our library consists of these 3 fluorophores, and it's still expanding, with the 4th fluorophore being in development right now. Our chromogenic offerings are growing as well, and with these chromogens, we're hoping to expand our library, enable researchers to give them decision whether or not they actually want to multiplex in fluorescence or in brightfield. And with some of these new these new chromogens that are coming out, we're hoping that they will actually exhibit the properties of fluorophores, and when they do co-localize, that they will form that third color. And I'll go a little bit over these chromogens in detail later. Okay, so before you begin to multiplex, first and foremost, you should definitely optimize each one of your primary antibodies using your most robust detection. And typically this is DAB. And DAB is tried and true. It's what the go-to detection that everybody goes to for a single IHC stain. Now, once you have your optimal assays for each one of these primaries, say you're trying to do a triple stain, what you're going to do is you're going to take each one of those antibodies that you have your optimal assay for, and then you're going to try and stain it individually with the color you're trying to hit it with. So say you have 3 biomarkers of interest, and you have 3 optimal assays for DAB. Now then follow up that optimal assay. Well, all you're doing is swapping out your chromogen, your DAB, with, say, a yellow. If you want your first biomarker to be yellow, and then your next biomarker to be in purple, and then your last biomarker, if you want to leave that with DAB, you can do so. And then see how each one of them performs individually before you actually throw them all together in a multiplex. Also, take into consideration the different optimal pretreatment conditions that will be necessary for different primaries. So if you have those same 3 biomarkers that I had listed in a previous example, but 2 of them require a less stringent or a less vigorous cell conditioning or pretreatment, then you would want to maybe find a happy medium for all 3, or detect the first ones with the less vigorous cell conditioning, detect those first, and then the ones that require a little bit more cell conditioning, you can detect those last. And then also, it's very helpful, at least to me, to map out what I'm doing. So if you're going to be dealing with 2 mice primary monoclonals and 1 rabbit monoclonal, just to map it out so that you can see, okay, hey, I'm going to detect this mouse primary with this mouse detection. I'm going to detect this rabbit with this rabbit detection. And that way, when you map it out, you can actually see where there is potential for false colocalization, false cross-reactivity. anything like that. So, you know, this signal should look yellow, and then I don't expect this antibody to co-localize with this antibody, so I shouldn't see these color co-localizations, that type of thing. When you map it out, it kind of just gives you a heads up of what to expect, what your signal should look like. Okay, so on to fluorescent multiplexing. One of the ways that you can multiplex with fluorescence is just using directly labeled primaries or secondaries. So the great thing about this is the fact that it's really low total assay time. So typically, if you have secondaries that are conjugated to fluorophores, that's great, but the one drawback of this is that you have to use primary antibodies generated in different species. So for this example here, we have 4 different, in this cartoon schematic here at the bottom of the slide, you have 4 different targets of interest, but in order to achieve this, you would have to have 4 different primary antibodies generated in 4 different species, and then you would come in with whatever these may be, maybe an anti-rat, an anti-rabbit, maybe an anti-goat, and an anti-mouse. And then the ability, you could cocktail all those primaries, put those down on the slide, have them incubate, and then you could cocktail all your secondaries as well, and then have them incubate, and thus cutting down on your overall incubation time. But not without its drawbacks in the fact that because you are not relying on enzymatic deposition, your signal may be a bit weaker than what you would expect to see with a DAB assay or a tyramide fluor assay. So just take that into consideration when using this type of approach for fluorescent multiplexing. If your signal is too weak, I mean, I did mention you can use enzymatic deposition with tyramide fluors. And typically this is done with HRP. So if you have your optimal DAB assay ready to go, it's real simple plug and play where you just remove DAB from the equation and you just come in with your tyramide fluor, and you already have your optimal IF assay. But the one thing you might want to consider is if, because it is enzymatic deposition, and fluorescence does tend to be a bit more sensitive, if you get such an intense signal, you maybe want to cut down on your primary antibody, maybe 1 to 2, with a 1 to 2 dilution than what you were using in a brightfield assay, if you were getting bleed through into other filters or other fluorescent channels during your interpretation. But when you are using enzymatic deposition of fluorophores, you will have to detect in sequence. It's just the nature of the beast. And one of the things that you can do, and the benefit of this is the fact that you can work with 4 rabbit monoclonals if you want to and detect them all in sequence. But you will have to elute the antibody that you just detected in between each round of detection. So unlike the previous method where you have the ability to cocktail and detect all at the same time, in this method you actually have to detect in sequence. And some of the methods you can incorporate, and which we've put to practice on our platform, is you can elute your detected antibody with heat. We do offer a denaturing agent as well, and the fact that it's just a mild acid solution. So you lower the pH on the slide in order to elute that antibody, affect the affinity of that primary that's bound, and elute it off the slide. What you can do as well, since these are enzymatically deposited with HRP, you can quench HRP just by using discovery inhibitor, which is a 3% hydrogen peroxide solution, which will quench any active HRP on the slide. You could also use other low pH solutions or even high salt solutions to affect the affinity of those primaries and get them off the slide so you can wash them. If you were actually to forego any of those previously mentioned methods, what would happen essentially is you come in with your first primary, And you would detect that primary with your secondary that's conjugated to an HRP, and then that would turn over your first fluorophore. And then you would come in with the second antibody that you're trying to detect. You come in with the respective anti-species polymer conjugate, HRP conjugate. But because you failed to elute that first sequence or quench any previously bound HRPs, you're driving a false colocalization. And the same goes for anything done in sequence after that, and the fact that you can see here where you're just ending up with just a mess of colors that shouldn't be deposited where they should be. So just to walk it through, if you were to actually do it with an elution step, you deposit your first round of fluors, which are covalently bound to the tissue. So when you actually do apply the heat or a low pH solution or any method to elute that antibody from that first sequence that you're detecting, your fluor remains, and it remains intact and non-bleached. And then you come in with your second fluor, or your second detection, and then you do an elution step again, and then your third antibody with that elution as well, and that turns over the same. Therefore, what is turning over is essentially depositing at where it should be. Some things to consider when using this type of multiplexing technique is the fact that there are no species limitations. So, as I mentioned, if you wanted to detect 4 rabbit monoclonals in sequence, you're at liberty to do so. Just be sure that you actually are getting a proper elution. Some antibodies, especially with some really high affinity antibodies, they are difficult to elute on the slide, and this would be a very difficult antibody to multiplex with. And if you do have these types of antibodies, just detect them last, so you don't have to worry about trying to elute them. When you are using enzymatic deposited fluorophores, they do generate a very intense signal. And like I had mentioned before, because these are enzymatically deposited, it's very easy to move from DAB into an IF or a fluorescent assay, where all you have to do is simply remove DAB out of the equation and plug in your respective tier mesh fluorophore. But because you are detecting in sequence, the assay times can get lengthy. And one thing to make note of is that Psi5 is oxidizable, or does get oxidized by hydrogen peroxide. So if you are going to be using Psi5 for your multiplexing in IF, just make sure that you are detecting it last. Okay, so here's an example of just using our fluorophores for a triple stain. You can see here the ability to separate them each from each channel. And just an example showing how one can easily transfer an assay from brightfield into fluorescence. And another example here, kind of highlighting the fact that you don't have to worry about species cross-reactivity because you are eluting antibodies in between detection steps. So here we have 2 rabbits being detected on the same slide in sequence with no cross-false colocalization. And here, the same, but with 2 mouse primary antibodies. So if your signal's too weak and you're using enzymatic deposited fluors, you can simply just increase the incubation time of the fluors on your tissue. As you can see here with this heat map, the longer you leave the fluors on the slide, the more turnover you have and the more deposition you have, the brighter your signal. You can also, maybe you are using less than optimal pretreatment times, so that is something to take into consideration as well. And you could also toy with concentrations of your primaries if you choose to. All right, so chromogenic multiplexing. When using brightfield, you can take pretty much the same approach that we discussed when in fluorescence. So if you have your primary and then your secondary enzyme conjugate, you can use DAB and utilize the umbrella effect that it has. And then when you come in with your second antibody of interest that you're trying to interrogate, and then turn over whatever chromogenic substrate you want, and then your third, and then that can turn over, in this example, a yellow chromogen. So if primary antibodies are all from different species, you can cocktail, but due to enzymatic deposition, you are going to want to do sequential detection. But it goes without saying, if they are from the same species, do not cocktail your primaries, and just go through the same motions that we discussed with multiplexing and fluorescence. Some things to make note of as well is just the umbrella effect with DAB. DAB, when it deposits, it tends to block off any other binding sites within the direct vicinity of your target. So if you have a mouse antibody that you're detecting first and you detect it with DAB, and then your next antibody is a mouse antibody as well, you won't get any false colocalization just because of the DAB creates this blocking effect of that first one. So that is something to utilize if you understand how that property works. Other color recommendations as well. Fast red is stable through denaturation. So if you want to heat the slide in order to elute your antibody, you can use it. Just be aware that fast red is soluble in alcohols and xylenes as well. If you are going to be using silver detection for IHC, it is oxidizable by it is oxidizable by hydrogen peroxide, but that is something that, much like Sci5, you just want to take that into consideration, and so detect it last. And with the purple detection that we have to offer, it is covalently bound to the tissue, and so once it's on, it's on. You don't have to worry about any loss in signal or anything like that from your elution techniques. All right, so as I mentioned before, in this example, we have the DAB, which is being detected first. And you can see here on the bottom row, when DAB is detected first, you don't have any false colocalization. So what's going on here is we have two mouse primaries, BCL2 and CD34. On the top, the red is deposited first. And on the bottom, the DAB is deposited first. And you can see here, when the red is deposited first, it fails to produce that umbrella effect. So when you detect your second primary, or you just throw on your second mouse detection, so in that middle column on the top, you see the CD34 red being detected by red, and then just following it up with mouse detection. So there's no second primary. You get like this brown blushiness on the slide, on the tissue. And then, however, on the bottom, when you detect the BCL2 with DAB first, and then you follow it by red, there is no room for the mouse detection that would generate the red signal to actually falsely bind to the first antibody, which in this instance is BCL2. So use that DAB to your advantage when multiplexing. So long as you're not trying to co-localize, DAB, that umbrella effect property of DAB can be very beneficial. So here you can see that the order of addition matters when you are detecting as well. So in this example, we have CD3 and CD8, and just with the biology that we know about these two is that all CD8 positive cells should be CD3 positive, but not necessarily all CD3 positive cells are CD8 positive. So with the umbrella effect and knowing that DAB has this umbrella effect property, you can see that when CD3 is detected in DAB first, you get no purple signal at all. You basically drown out or you've blocked off any type of CD8 binding. But when CD3 is in purple first, you could see some sort of DAB popping through, but it doesn't look as crisp. So when you do a color swap, actually, when you detect CD8 first, the DAB really pops through, and then you can actually detect all your CD3s. And then when you do CD8 in purple, and then follow it up with CD3 and DAB, you can actually see the colocalization there because of the actual purple not having that umbrella effect that DAB has, and knowing that all CD8s are a subset of CD3s, you can actually see the colocalizations in these ones. Here's just a triple stain chromogenic example that we have. In this, you wouldn't need to worry about the umbrella effect, just for the fact that you're staining 3 different cellular locales here: membrane, cytoplasmic, and nuclear. And the beauty of using these 3 chromogens in this particular example is the fact that they are covalently bound to the tissue, so there is no need or no worry that they will disappear or dissolve in your dehydration station. These chromogens are in fact highly stable in alcohols and xylenes. Okay, and this just another example to highlight how you can tease apart the differences between our purple kit, which is an HRP substrate, and our red kit, which is an alpha substrate. All right. So in closing, I hope that I was able to at least convey the message that fluorophores are, while being highly advantageous, the fact that they can co-localize and you can do a sequential enzymatic deposition with them for multiplexing currently. However, they're not very stable. You can't really archive them, and they do require special microscopes in order to interpret your results. And the same goes with the chromogens, and the fact that they don't, at least currently, chromogens don't have the properties that fluorophores do, and the fact that when they colocalize, they tend not to form third color, because all we really had currently has been DAB, and brown mixed with any other color is just going to be a slightly different, uglier color of brown. So it's really not beneficial to multiplex, or it hasn't been up until these new colors have been coming out. So what we're hoping is that we are developing colors that maintain the properties of fluorescence, so that when they actually do colocalize, they form that third color. And so the chromogens behave like fluors in that aspect, but they don't behave like fluors in the fact that you can't archive them. So and who's to say that maybe there could be an opportunity that we can actually shorten the assay time by utilizing these chromogens and then depositing them at the same time, like you can do with fluors that are conjugated to secondaries. So if you're using a chromogen that is an HRP substrate and a chromogen that's an alpha substrate, and then you lay them down at the same time, you might be able to cut down on your assay times that way as well. So here is just a couple of examples that we have where you have yellow, our new chromogen. Unlike any other yellow chromogen out there on the market, is the fact that it's highly stable. You don't have to dilute it out and then use within 48 hours. It's fully automated, up and running on our platform. And like as mentioned before, there's no interruption in workflow. You could treat this slide that you stain with yellow and purple, for that fact, just like you would treat a DAB stain slide. And then here, just kind of a snapshot into some of the other chromogens that we're working on that also have that same property of no interruption in workflow and highly stable. As you can see here, colocalization, so that when you actually do have a yellow and a purple colocalized, you get an orange color, or if you have a yellow and a teal color, you create like this green color. So these are things that we are working on here at Ventana, where we're trying to capture the benefits of a fluorescent assay, but bring them into the brightfield space. So with that, I thank you for your time, and I'd be happy to field any questions.
Great. Thanks, Adrian. That was a great presentation. You showed us some really great images, as well as some new staining techniques for multiplexing assays, so we really appreciate that. Thank you. So before we move on to Ed's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions at the final presentation, or after the final presentation, excuse me. So let's move on. Ed, as they say, the floor is now yours.
My name is Abril. I am at the University of Arizona Cancer Center. I work in a tissue core lab that does exclusively IHC for our investigators. And the title of my talk is Chromogenic and Multiplexing Staining using the Ventana automated platform. I just want to give you a brief introduction into immunohistochemistry. We should all know what immunohistochemistry does. It basically detects a protein of interest using an antibody. And this is visualized using DABMap, which it has been a standard for a long time. Sample preparation is very important to me. I have investigators that will bring me samples that are not prepared properly. And if they're not prepared properly, as the old adage goes, garbage in, garbage out. So I have to talk with my investigators and make them aware that the sample preparation is important. So when I get a good sample from my investigators, I have to choose a proper tissue control that is a high expressor for the antigen of interest. Now, I can go to several databases. One is the Human Protein Atlas, and that one gives me a good idea of what control tissues I can use that express the antigen of interest that I'm looking for. The second step that I go through is to determine correct antigen retrieval condition. I have 5 antigen retrieval conditions that I have to go through to determine which one is going to demonstrate whether it's good nuclear staining, good cytoplasmic staining, or both cytoplasmic and nuclear in some cases. My next step, which is very important, is I have to determine an antibody titer. Once I've determined an antibody titer that shows proper staining, both intensity and location, then I am free to go to using the Chromogenic kits. And the first Chromogenic kit that I will use is a DABMap. I will take the antigen retrieval conditions and the antibody titer that I have determined, use the DABMap, look at the control tissue, and once I have determined that everything is correct, I will then, if it's for multiplexing, I'll either use a blue, red, or purple map kit. And here I have to be careful when I'm co-localizing. If I have a localized protein, that color doesn't come out, burnt orange, for example, that it's a color that can be easily visualized. If I have a protein that is relatively low expressing, I will use the Ultramap HRP. That then I will have to use make stop. If I have a low-expressing protein, I use Ultramap, and I use the same conditions that I did in determining what antigen retrieval conditions are correct and antibody titer is correct. Next, the types of detection. There are multimers that Ventana has. These are long armed linkers that attach the enzyme directly to the secondary antibody. And so these are invaluable to me because I hand apply both my primary and sometimes my secondary if the case calls for it. There are smaller Multimer molecules that minimize steric hindrance, and they improve the sensitivity while keeping a biotin-free system in place. Next, I will use the DABMap Kit, which is the standard that I use for everything before I even begin to think about multiplexing. The DABMap Kit relies on an interaction between HRP and the SA complex and biotinylated antibody and the reaction with the DAB, which produces the brown staining. Here's an example of prostate cancer with a beta-4 integrin antibody, and as you can see, the basement membrane is stained along with the epithelial cells in this particular prostate cancer. Ultramap use in conjunction with the DAB, as I've said previously, if these are low-expressing proteins, because low-expressing proteins do not come out very well with DABs. They either show a blush or they show no staining at all. And here are three examples of Ultramap HRP DAB staining on your upper left-hand side. You'll see Ultramap DAB. On your right side, you will see HRP red, and at the bottom you'll see HRP purple. One other agent that is very important to me is the NPAP system from Ventana. It's a mouse monoclonal antibody. It's designed for haptenated NPs, and it's conjugated to an alkaline phosphatase that can be turned over for kits such as the Red Map Kit. The BCI is found in the Discovery Chromogenic Blue Kit. The anti-NPAP provides a very clean background with a very low signal to a very low signal to background. And here are two examples of this. The NPAP red kits, which is clearly shown on the left-hand side, and on the right side you have an NP-AP kit with a low-expressing protein, so you are not seeing the red as clearly as you are on the left-hand side. Let me get into why I use the DABMap Kit. It relies on an interaction between HRP and the assay complex. You use a biostimulated secondary antibody and you get a brown reaction. Here is an example of prostate cancer with a very low expressing antibody, plectin, which is an integrin molecule needed for invasion of prostate cancer further into the prostate gland. And since this is a low expressing protein, this is a good example of why I have to use Ultramap Kit, because the DAB does not bring out the plectin in prostates or some of the other tissues that I have used. Here is another example of a low-expressing protein using DABMap Kit and ERG. And as you can see, there is very little brown staining because it is a low-expressing protein, where on this particular tumor, when Ultramap was used, we did see a noticeable difference. Multiplexing is been used in genomics, and it's been used to detect biomolecules, both protein and genes in tissue. This allows you to determine the effect of a specific treatment that you're using, or to see if this antigen or DNA is expressed in your tissue. It also allows for protein-protein interactions. Here is an example of a multiplexing picture with RedMap kit and purple and blue counter stain. My next slide is a prostate cancer picture where I am using the same colors, and I'm also using DAB, which in the previous slide you did not see. Here you can clearly see the DAB, the red, and the blue map kit in this particular multiplexing example. Multiplexing methods, in order to achieve good multiplexing results, you need to consider where the antigen is. Where is it localized? Is it in the Golgi? Is it cytoplasmic? Is it nuclear? Is it in some organelle? Then you need to determine are you going to have co-localization? If you're going to have co-localization, you need to choose the colors that you are going to use very carefully. If you're going to use red map first, and then you're going to use blue map next, that's going to give you a color that you can work with. If you use a red map and a DABMap kit, the DABMap kit is going to overwhelm the red map kit. So for co-localization experiments, all of this needs to be mapped out to eliminate unwanted and staining, color staining. The assay protocols must be optimized for each antibody. When you build the protocols, they must be stacked in an order that both accounts for the color kits that you're using and the species of antibody, of your primary antibody. So, for example, if you have a rabbit primary and you're going to use red, you cannot use another rabbit antibody for your second stain, which, for example, would be the Ultramap. What you need to do is to have, either 3 different species, which is hard to find, or the best case scenario is to have a rabbit, a mouse, and a rabbit, and this will give you good staining if you have picked out the right color chromogens, and they do not give you any weird staining. In conclusion, single colorimetric staining is useful for single antigens, but once you've established the conditions for 3 antibodies or 4 antibodies, you need to design one protocol that's designed takes into account all those parameters. Of great importance, and I can't stress this enough, you need to pick out your proper colors, put them in an order that that is going to give you a readable color. If you don't, you will have wasted, anywhere from 6 to 12 hours in doing nothing. And in conclusion, I'd like to thank Ventana Medical Systems, a member of the Roche Group, for providing me with continuing training and classes. The customer support is excellent. If I have any questions or I run into difficult problems, which I have in the past, and I've called the technical application specialist up, they will point me in the right direction. And sometimes for some of the projects that I have run into, I have stumped them. So it takes the 2 of us, both Ventana and myself, working on the problem to resolve the problem for the investigator to be happy with the results. I'd like to also thank Genetic Engineering News for putting together the webinar, and thank you for this opportunity for presenting.
Great, thanks, Ed. That was really excellent talk. You really showed us some nice images and techniques, in addition to presenting us with the versatility of the Discovery Ultra platform. So we thank you for that. So before we start the Q&A session, I just want to let everyone know this is your final chance to submit your questions for our speakers. So hurry up and submit them now. All right, let's begin the Q&A. I see a few questions already starting to roll in, so why don't we start answering a few of them, and we'll try to get to as many of them as we can. Okay, so let's get to our questions. We have a few coming in. They look pretty good. Adrian, the first one's going to be for you, and it's actually our audience member had a comment that said, for multiplexing, it's not absolutely necessary to use primary antibodies raised in different species. Instead, primary antibodies raised in the same species can be modified, conjugating to biotin, deoxyribogenin, and other tags, followed by anti-tags and secondary antibodies. So I thought maybe this was something that you want to comment on.
Yeah, actually, that is another method for multiplexing that one can utilize. I didn't quite go over this just for time constraints, But yes, you can definitely hapten label your primaries and detect them with the respective anti-haptens. Just some things to consider when you do incorporate this technique is the fact that when you are modifying your primary antibodies with these tags, just be careful because you may compromise the affinity binding region of these primary antibodies, and also you may be compromising the tertiary structure of these antibodies and thus affecting their overall stability. So there's just a couple things to consider. Some antibodies perform great once they're hapten labeled. Some other antibodies do not. So it's all a matter of trial and error with this type of technique.
Great. Thanks very much, Adrian. So, Ed, we have a question for you. One of the audience members wants to know, why is multiplexing important?
Well, multiplexing is important from the standpoint of view that in tissue, if you look at just a regular DAB stain or a single stain, it only gives you a limited amount of information. If you are looking at, if you're interested in looking at two antibodies that are in the same pathway, for example, and they work in connection with each other, it is good to look at the tissue and actually see that those two proteins are being expressed in the tissue. Whether one is expressed more than the other, that comes out on the IHC, and that is very useful in determining your results. And that's why I would say multiplexing is important. If you get into three-color multiplexing, again, you have to you're looking at a pathway that is very integrated, and the more integrated a pathway is, you want to look at how those specific proteins are interacting with one another.
All right, thanks, Ed. Adrian, we're going to go back to you. So one of our audience members wants to know is, do you experience any loss in signal due to heating of the slides or reagents that you apply to elute previously bound antibodies?
I have not observed any loss in signal with some of the fluorophores that we have, and also with the chromogens as well. With the chromogens that are novel to Ventana, like I had mentioned, they are covalently bound to the tissue, and we have yet to see or hear of any experiences in which any type of elution step, whether it's heat denaturation or applying a mild acid solution to the slide, is detrimental to the signal generated by these new colors and fluorophores. Aside from the examples that I mentioned, where the silver and the Psi-5 both are affected by prolonged exposure to concentrated solutions of hydrogen peroxide, other than that, we have not seen or experienced or observed any detriment to signal generated.
Okay, great. Thanks, Adrian. Ed, we have another question for you. One of the audience members wants to know, what are some of the problems that researchers are faced with when performing multiplexing staining?
Some of the problems that researchers come across is, for one, the proteins are so co-localized that even if you use different color detection kits, the color that you get is so either bleached out or so overwhelming that it overwhelms the other two colors, and it is virtually useless to determine anything from the tissue that you're looking at. Another problem is multiplexing, even though it does have its advantages, the lack of a broader spectrum of chromogens, specifically the types that I use, the purple map kits, the red map, etc. A broader spectrum would help for the simple reason that you would get you could do a broader range of proteins, and with the limited chromogens that we have, you are very limited to what you can do if the proteins are, like I said, are relevantly or covalently bound to each other or in some way linked to each other, and use a yellow map kit or a purple map kit, or one of the other kits, you may not get a readable result from doing that type of experiment. So in that case, what I would do is I would do serial sections and just stain one serial section with one color kit and the other with the other color kit, and then just go and microscopically overlay the images, and then with filters see if I couldn't clean that image up.
Okay, great. Thanks, Ed. So, Adrian, we have another question for you. Would it be possible to do a combination of direct labeled fluors with enzymatic deposition of fluors?
Yeah, that definitely would be feasible. The one recommendation I have, if one were to attempt this, is definitely use the enzymatic deposited fluors first. That way, they're not affected by your elution methods. And then, if you're going to be coming in with secondaries, if they're anti-hapten secondaries or anti-species secondaries, if your elutions were effective, there shouldn't be any cross-reactivity going on there. So you can then throw down your primaries of interest and then your respective anti-species secondaries that are conjugated to fluors. So I guess, so if you're going to do that, do the enzymatic deposition of fluors first, followed by the anti-species fluor conjugates. And thanks.
All right. Great. Thanks, Adrian. So it looks like we have time for one last question for you, Ed. One of our audience members just wants to know, what's the typical length of time for a multiplexing assay?
That can range from anywhere from 6 hours to 8 hours, depending upon how many fluors are being used. If I'm using the maximum amount of fluors, it could be an 8-hour experiment.
Okay, great. Thanks, Ed. And I think with that, we've come to the end of our webinar. I just want to remind everyone that the webinar will be archived for 6 months on our website. So if you missed parts of it or you want to watch it again, you can feel free to do so, or you can forward it to your friends or colleagues, which we always recommend. I'd like to thank Ed and Adrian again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Roche for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.